Let's see. A reminder that quiz 2 is due tomorrow at 4:30 p.m. Most of you still have to take it, so please don't forget. Then another reminder is exam 1, the first exam, is on Tuesday after break, so a week from today. And there are some options, and which I put on Moodle. I'll tell you now. So the first option is to take the exam here during the normal time, right, 8:30 in the morning on Tuesday. That's an option. Because there is no lab on Tuesday, you can take it, an option is to take it at 1 o'clock or at 3 o'clock on Tuesday. So those are options available, and you don't have to tell me, you just have to take one of those options. And I don't want to, you know, if you say, oh, I'd like to take it at 1:30, say, come on, just take it at 1 o'clock. I mean, I would rather have a finite number of start times rather than an infinite number of start times on Tuesday. If you are taking the Econal exam on Tuesday, if that is a conflict for you, then there are some additional options on Wednesday. Okay? I'll make the exam available to those of you who are in Econal and have the Tuesday exam time on Tuesday. You can take it on Wednesday at 9 in the morning or Wednesday at 1 in the afternoon. But that's only if you have the Econal conflict on Tuesday. If you don't have the Econal conflict on Tuesday, I want you to take it one of those Tuesday times. Okay? Clear? Today what we're going to do is talk about hippocampal long-term potentiation. This is the starting point for the formation of declarative memory, which hopefully you all are forming readily, frequently, continuously during this class. Okay, so we're going to talk about the starting point for the formation of memory and use that as a springboard to remind you of and answer questions about some of the key concepts for this section of the course. Okay, so it's serving two purposes. One is to teach you about learning, the molecular mechanisms of learning a declarative memory. Declarative memory is, you know, memory of facts and names and you know, events. That's the kind of memory you're using in this class. Procedural memory is the other basic type of memory, which is motor memory. Learning how to ride a bicycle, learning how to throw a frisbee. Different. It's actually got a different molecular mechanism. Declarative memory depends on the hippocampus. If your hippocampus is damaged, you stop being able to form memories. You don't lose existing memories, you stop being able to form new memories. One final announcement before we do that, before we start going into hippocampal LTP and the formation of memories, is just an announcement that next Thursday, the Thursday after break, there will be an organizational meeting for the Brain Explorers outreach event that will be happening in March. And so the organizational meeting to talk about, you know, what are some of the options will be, you know, during the biology colloquium slide on Thursday, February 21st. Here. Question? Yeah.
Choose whether participating versus station will give extra credit?
Both will give extra credit. I don't know about equal, but both, it depends. So if you, both will generate extra credit for the course. If you come to the Brain Explorers event and engage fully, you will get extra credit. If you generate a station and you participate fully, it will generate extra credit. Approximately, not a huge difference. Yeah?
I'll schedule it for the lab this week, but does the Tuesday lab have?
No, there's no lab next week.
Okay.
There is Tuesday lab this week.
Okay, I just want to make sure.
Did I get that wrong? There is lab this week. There is no lab next week. No, let me make that clear. There is Tuesday lab this week. There is no Thursday lab this week because it's break. There is no Tuesday lab next week to make up for the fact that the Thursday, to keep things aligned. And there is Thursday lab next week. Okay. All right, hippocampal LTP. Hippocampus is a section of the brain. This is a cutaway view of the temporal lobe from the side. It's a little funny orientation, but we're looking at a lateral view of the brain with a cutaway of the temporal lobe, looking into the deeper structures of the temporal lobe. So this is, the hippocampus is in the medial temporal lobe. So when you look at it from the side, you have to cut away the lateral temporal lobe in order to see the medial temporal lobe. It's called the hippocampus. If you dissect it out, it looks something like this. The Greek word for seahorse is hippocampus. That's where it gets its name. So here is a coronal view of the brain. Medial temporal lobe. So temporal lobe, medial, there's the hippocampus in this perspective. Okay? Now I could remind you that this kind of view is fair game for both quiz 2 and exam 1. Okay, so being able to identify structures. And we've already played the naming game, so I'm not going to play it again because I want to move on. But you could use this slide, which will be on Moodle, or it's on a previous handout, or at least something like it is on a previous handout, to label, to see if you can label. You won't be able to label all 38 lines here because I didn't teach that, and you didn't learn that. But you can label many of them. Thalamus, lateral ventricle, corpus callosum. I'm doing what I said I wasn't going to do. You do this. So if you take the hippocampus, let me just remind, so here's the hippocampus, and it's got this sort of interesting kind of spiral shape. And you slice it in a specific orientation, and this is a rat hippocampus because that's the typical experimental approach to understanding learning and memory. We don't do very many human hippocampus experiments. We do mostly animal hippocampus experiments. This is a rat hippocampus, and it has this very defined cellular structure. So these dots are cell bodies, and the purple haze is the location of axons and synapses. So soma in the dentate gyrus, and soma in the so-called CA3, CA2, and CA1 region of the hippocampus. And in here are the axons and synapses of these cells, whose cell bodies are labeled. And here's a cartoon of the same structure. The cell bodies of the dentate gyrus, their axons project to the CA3 region and form synapses on the cells of the CA3. Here are the dendrites of the CA3 cells, and here are the axons of the CA3 cells that form synapses on the CA1 cells. And this is a highly ordered, layered sort of structure that is reproducible, and if you slice it right, you get something like this. This synapse, the synapse between CA3 neurons and CA1 neurons, is the synapse that is the most characterized synapse in the world, I think, because it's the source of long-term potentiation, which is the cellular and molecular manifestation of the formation of memory. When you form a memory, there is potentiation of this synapse. Potentiation means it is stronger. You get a larger EPSP after the formation of memory than before the formation of memory. This synapse strengthens, potentiates during the formation of memory. So there's input to the hippocampus, and that input is, electrical activity. That electrical activity generates electrical activity in the CA3. That electrical activity in the axons of the CA3 neuron potentiates the CA1 neuron, and then there's output as well to the cortex. So we're focusing on the potentiation of this synapse. Axons making synaptic terminals on the dendrites of these CA1 neurons. That's the synapse that that's one of the synapses that potentiates during the formation of memory. And this is an example. So here is a misspelling of synaptic response. This is synaptic response normalized to the starting point. So the synapse was stable. The EPSPs of that synapse were stable before the stimulus. And then a stimulus was given to mimic the formation, the introduction of a new fact, right? When you are learning a new fact, there's a buzz of electrical activity that causes short and long-term potentiation of this synapse. After the synapse, after the learning, after the stimulus, those synapses are different than they were before. So when you learn a fact that bar across the coronal section is called the corpus callosum. That name is now embedded in your memory, and when that memory formed, your brain changed. The synapses in your hippocampus changed. And then that information was processed to your cortex where other changes happened. But the first step was the potentiation of that synapse in your hippocampus. So we know, and there's really good evidence, that this phenomenon of long-term potentiation, long-term just means, in this case, it's only lasting an hour. You know that memories last longer than an hour. I hope you know that memories last longer than an hour. I hope your memories are lasting longer than 1 hour, because it can go on for days or weeks or years. Pretty much as long as we can measure. So now let's use the cartoon of that synapse as a model for learning, for review of the key concepts of this course so far. So let's get oriented. This is the presynaptic terminal of the CA3 neuron, right? This is the presynaptic terminal of this axon. And this is the postsynaptic spine of the dendrite of the CA3 neuron. So that's CA1, this is CA3. Presynaptic, postsynaptic. So let's talk about some of the things that we can review here. I don't want to reteach it. There's no way I can reteach, 3 or 3.5 weeks of the class in 45 minutes. I don't want to even try. But what I want to do is mention key concepts. And if you have a question, now would be a good time to bring it up. So these, so this is supposed to be the presynaptic terminal. These are supposed to be synaptic vesicles loaded with the neurotransmitter glutamate. Well, okay, so what are the concepts? Well, you know that this cell has a resting membrane potential. And you should know something about the molecular mechanisms for the resting membrane potential, the ion gradients that were formed, the channels that are open or not open that cause a negative resting membrane potential. So just mention resting membrane potential. An action potential invades that synapse. So now I've reminded you that knowing the molecular mechanisms of an action potential, what channels open, what channels are not open, at what phase of the action potential, when are sodium channels active, inactive, and resting closed. Action potentials invade this terminal. Voltage-gated calcium channels here trigger neurotransmitter release. The molecular mechanisms of neurotransmitter release. Calcium coming through voltage-gated calcium channels, binding to synaptotagmin, conformational changes of the T-snare, V-snare complex, fusion of vesicles and release of neurotransmitters. But I could also ask you, okay, after a neurotransmitter is released, how does it get back, how does it get recycled, re-uptaked, taken back up into the presynaptic terminal and reloaded into synaptic vesicles? What are the molecular mechanisms for reusing the glutamate that was previously released from a previous release event? So those molecular mechanisms of neurotransmitter reuptake. Glutamate as a neurotransmitter, excitatory. Glutamate receptors. So what type of glutamate receptor is being represented here? Ionotropic glutamate receptors. They're looking like channels. This is a donut cutting cross-section. Ions are moving through. These are ionotropic receptors, or ligand-gated ion channels. And so an excitatory amino acid, an excitatory neurotransmitter acting through ion channels that are sodium and potassium permeable, like the acetylcholine receptor that we talked about that you did on a problem set, would cause a depolarization, sodium influx and depolarization of the postsynaptic cell. That's an EPSP. So I just mentioned EPSP. Any questions about any of these key words or the words that I just said in the past 3 minutes? Because those are key concepts that we've been using so far in this course, and will continue to use. Okay, so now let me talk. There are 2 types of glutamate receptors in this postsynaptic cell. And I'm not going to test you on the 2 types. I've mentioned them already. There are the so-called AMPA receptors and the so-called NMDA receptors. And they're named for pharmacological reagents that activate them. AMPA is a chemical that is a glutamate analog that specifically activates a subtype of the glutamate receptor. Now, this is just a fact that I don't expect you to know for the exam, but the NMDA receptors, when this cell is relatively polarized for extended periods of time, this receptor is blocked by magnesium. It's got a voltage-dependent block by an ion that is present in the extracellular solution. So when this cell is negative, magnesium it pops into this channel and blocks it and prevents ions from flowing through. But these channels are not blocked by magnesium, and so the neurotransmission, glutamatergic neurotransmission, is maintained through the AMPA receptors. Glutamate is released, AMPA receptors open, sodium flows in, depolarization, yada yada. But if there is a large amount of glutamate release, like a new fact is being learned, a new stimulus is going into the hippocampus, a large amount of neurotransmitter, high frequency stimulation, this cell becomes depolarized enough for long enough to kick the magnesium out of the NMDA receptor. So what I've just talked about is voltage-dependent block and unblock of the NMDA receptor. At regular voltages, it's blocked. After a high frequency, large stimulus, it becomes unblocked. When this channel becomes unblocked, NMDA receptors, in addition to allowing sodium to flow, potentiating the depolarization, these also allow calcium to flow. Calcium goes in. In this case, so calcium now binds to calmodulin. I don't know where the E came from. Calcium binds to calmodulin and activates things. A calmodulin-dependent protein kinase called CAM kinase 2, calcium calmodulin kinase, and adenylocyclase. So now this postsynaptic spine is changing, right? Things are gonna happen because kinases and creators of second messengers have been activated by the calcium that came through the unblocked NMDA receptor. And they were unblocked because they just got a buzz of activity. So what we just talked about is how a large depolarization led to unblock of NMDA, voltage-dependent unblock of NMDA receptors, calcium as a signaling event. Now we're getting back into generic. What is another way that you should be thinking of? When I say calcium went up here, you should say, oh, that's just like what would happen if we activated a GQ-coupled metabotropic receptor. But this isn't a GQ-coupled metabotropic receptor. It's a NMDA subtype of glutamatergic ionotropic receptor. Calcium can go up in cells in any number of ways. They can do a number of things. You've probably learned calcium activates protein kinase C. Yes, it does. It also activates calmodulin and CAM kinase 2, another kinase. So I'm using some of the concepts you've learned to teach some new stuff, but the increment is relatively small. Calcium is a major signaling event. Calcium, calmodulin-dependent protein kinase, and the adenylyl cyclase. This kinase phosphorylates AMPA receptors that are present in the cell and gets them into the synapse, puts them in the synapse. So this phosphorylation increases the number of AMPA receptors. What's that going to do to the size of the EPSP the next time a normal release of glutamate happens? It's going to increase it. More receptors, even if this one has become blocked again, because now we're back to normal, but more receptors means a higher EPSD. And this can last for a long time. Long-term potentiation, right there. Putting that another set of AMPA receptors, because they were phosphorylated by CAM kinase 2, increases their expression and insertion in the membrane, which increases the response to a given amount of glutamate. New receptors, bigger EPSP. Right there, long-term potentiation. In addition, now here's a story you know. What does adenylyl cyclase do? Make cyclic AMP from ATP. Cyclic AMP activates protein kinase A. This is the GS story, but we didn't have to activate GS. We activated calmodulin. PKA increases, phosphorylates a transcription factor. The genes of this postsynaptic cell change. So this information is transported all the way back to the nucleus, and more proteins get synthesized. Protein synthesis, not just the insertion of previously synthesized AMPA receptors, but the synthesis of new proteins, and in fact, building of new spines, new synapses, makes for these three things: insertion of new AMPA receptors, transcription of genes, increasing the size and number of synapses, causes the long-term change in the hippocampus that is responsible for learning, for LTP. So I've just told you a new story, but I've used some of the key concepts that you should be familiar with in reminding yourself of. Leah. No. If we have just a normal action potential that's happening every second or so, just sort of a steady rate of information flow through the synapse, that's not enough to kick this magnesium out. It's only when we get a high frequency, large amount, like hundreds per second, high frequency, a new event, I'm waving my hands because now we're talking about, all kinds of complicated input to a simple synapse. But simple throughput does not release enough glutamate to kick this magnesium out. It's the buzz of high frequency stimulus that does enough depolarization to kick the magnesium out.
And also for the AMPA.
Oh no, there's AMPA receptors and there's NMDA receptors throughout the brain and the spinal cord.
So you're saying this doesn't just happen at that
Oh, so no, this does not just happen at the synapse. This is the best example of LTP. But we can have LTP at other synapses. So for example, drugs of addiction like cocaine or opiates do something similar in a different part of the brain. So let me tell this story briefly, but not in quite so much detail. So this is a cell body, dendrites, axon. In the ventral tegmental area, the VTA is one of those homes of the cell bodies of dopaminergic neurons. So we're not in the hippocampus anymore. We're in the VTA of the midbrain, the ventral tegmental area. Dopamine is the neurotransmitter there, not glutamate. Dopamine activates dopamine receptors. Normal pathway. Dopamine receptors are coupled, some of them, D1 receptors are coupled to GS, and now we're running on the DS story. DS. All kinds of things that you should know about before we activate adenylyl cyclase. I'm not teaching it again. This D1 receptor, many things, adenylyl cyclase, ATP being converted to cyclic AMP by adenylyl cyclase, PKA, we're seeing the same story here as before, PKA, Krebs, changes in gene transcription of the target cell, which is in the nucleus accumbens. That's what NA stands for. Nucleus accumbens, that's one of the targets of the dopaminergic cells whose cell bodies is in the ventral tegmental area. These cells change if there is enough activation. Cocaine, now we're getting into the detail that I don't expect you to memorize. Cocaine is an inhibitor of the dopamine reuptake transporter. Remember, now I'm mentioning something that I do want you to know. The molecular mechanisms for dopamine reuptake from the synapse. Know it. Don't memorize that cocaine is an inhibitor of it. But now think, if we inhibit dopamine reuptake, it stays in the synapse longer. Higher concentration of dopamine in the synapse longer, more activation of D1, more activation of this pathway, more gene transcription, changing your brain in response to a drug of addiction. We could talk about this one. Here's a glutamatergic synapse. Glutamate as an excitatory neurotransmitter here. What do you suppose EAAR stands for? What is glutamate as a chemical? It's a what, Mackenzie? Excitatory amino acid. There it is. Excitatory amino. So this is AMPA and NMDA receptors. Excitatory amino acid receptors. And so we can have other things from the prefrontal cortex and from the amygdala and from the hippocampus can cause potentiation of this neuron. So you can learn to be rewarded by an event. Anyway, the point is learning from hippocampus, fear memories or emotional memories from the amygdala, things from the prefrontal cortex can cause changes in the midbrain that can lead to changes in behavior. that's what we're talking about. What we're leading up to is addiction, which is a very complicated result of changes in this. This is the starting point of addiction. Okay, so I've used these two interesting stories. Hopefully they're interesting. I've used these two interesting stories to try to mention some of the key concepts. Let's do a bunch of clickers. We've got time to do a whole bunch of clickers that are, again, using the concepts. My goal is not to quiz you. My goal is to get a conversation going here about these key concepts. I think the best answer is these three. Dopamine has to diffuse around its cleft. It has to bind to the sodium dopamine cotransporter, DAT. It has to get there, so it has to diffuse there. It binds to it along with sodium. Sodium and dopamine bind to DAT. DAT uses the sodium gradient to pump dopamine into the cell. That sodium gradient came from the sodium potassium ATPase. Okay? Same question? Different answers. Dopamine is not a neurotransmitter that is hydrolyzed. Acetylcholine is, but not dopamine. Circa transports calcium, but none of this process is calcium dependent. It's a pumping of dopamine from the cytosol into the vesicle depends on a proton gradient established by the proton ATPase and a exchanger process in that membrane. Questions about these concepts of creation of gradients, neurotransmitter uptake, and process, packaging? Okay, let's go on. Giving you some new information in the stem of the question, and then the branches ask you to apply your knowledge about this terminology. I don't care that you learn about tubocararine, but you should know what high affinity, competitive, antagonist, what those words mean. You should know what the acetylcholine receptor at the neuromuscular junction is. That's the nicotinic acetylcholine receptor. So now I'm asking you to apply your knowledge of what those words mean to answer these questions. Now I'm asking you to remember what a KD is. I'm asking you to remember what an apparent KD is in the presence of a competitive antagonist. I'm asked, yeah, those are, so now show me your answers. I think they're all right. So let's go through them. A high affinity means a low KD. Competition means that in the presence of the competitive antagonist, the apparent KD is higher, but the apparent max is unchanged. That's the definition of a competition. So you have to remember, relearn by next Tuesday, what a competitive versus a non-competitive inhibitor does to a receptor. This is competition. They bind to the same site. And now, okay, this one, we haven't gotten into, well, we've talked about the neuromuscular synapse, right? If we block the acetylcholine receptor with a competitive antagonist, we're gonna block neurotransmission to the muscle, and so the muscle's not gonna contract well, and that'll cause weakness or paralysis. That's how tubocurarine works. This is another toxin that's used by indigenous American Indians to, as a poison, a dart poison, to paralyze their prey. Questions about any of these words? What I want you to know and what I don't want you to know. Okay.
We previously looked at like paralysis when you have like overactivation.
So there's two types of paralysis. We can paralyze by having extended paralysis, where I can't flex my elbow if the extensor is hyperactive. I need to be able to relax my extensor in order to be able to flex my elbow. So that requires contraction and relaxation. So this is flaccid paralysis. I can't contract the muscle because the muscle is weak. The other type of paralysis we've talked about is spastic paralysis, where I can't relax the antagonist muscle. Both of those cause paralysis. Okay. Activation of which of the following is associated with a downstream increase in cytoplasmic calcium? Now you have multiple choices. Yes, no, and maybe, or sometimes. So each blank has one letter, so we're just going to go through them one at a time. So show me one letter. For GI coupled GPCRs. Yes, no, or maybe. Yes, no, or sometimes. I can't remember what I put here. I think, no. GI coupled receptors are coupled to decreases in adenylyl cyclase and increases in potassium permeability. Both of those are inhibitory, but that doesn't lead to, we have to stretch in order to get to a change in cytoplasmic calcium. I can see, an argument could be made for B. I will admit. Okay, GQ coupled GPCRs. This one's pretty straightforward, right? Yes, right? This one is well known to cause an increase in cytoplasmic calcium. GS coupled GPCRs. I think the best answer here is B. Sometimes, or maybe. Because it depends, remember, GS coupled, what is the canonical result of an activation of a GS coupled receptor? Activation of adenylyl cyclase, cyclic AMP, PKA. What if PKA were to activate SERCA? Would that cause an increase in cytoplasmic calcium? No, that would cause a decrease in cytoplasmic calcium because SERCA pumps calcium into the SR. What if pKa activated, phosphorylated and activated voltage-dependent calcium channels? Would that cause an increase in intracellular cytoplasmic calcium? Yes, now we have increased an influx of calcium, and that could. So those are the two, what I just taught you is a key concept from AMP2, that activation of GS-coupled receptors can cause either an increase in cytoplasmic calcium due to phosphorylation of calcium channels, or a decrease in cytoplasmic calcium due to phosphorylation of SERCA. So that's a yes, no, I mean that's a maybe, or sometimes, depends on the cell. Nicotinic ACH receptors. Now you have to apply what you know about ionotropic nicotinic ACH receptors. Do they cause an increase in cytoplasmic calcium? They do, because they are calcium permeable. They are like NMDA receptors that once they are unblocked from their magnesium. Oh, what about NMDA receptors? Okay, I just leave that to you. What about ionotropic GABA receptors? Many of you are not showing me your answers. Does that mean you don't know, or because you're just too sleepy? No. This one would be a no. What do ionotropic GABA receptors do? Chloride. What is that? Is that excitatory or inhibitory? Inhibitory. So we have to really stretch to get from influx of chloride and hyperpolarization to get to an increase in cytoplasmic calcium. I'm not sure I could get there. Okay, so being able to apply these things, thinking about what happens in these kinds of receptors, is learning about the brain and learning the molecular mechanisms of brain, of these events that we've been talking about. Okay, let's do another one, and then we'll take a break. Which of the following will usually cause a hyperpolarization? Right, hyperpolarization, only C. Let's go backwards. Or let's go from the top. What would a high potassium concentration in the extracellular solution do? What if this neuron was being bathed in an abnormally high potassium concentration? It actually happens during epilepsy. It's going to cause a depolarization because the potassium gradient has now been collapsed a little bit, and the potassium gradient was what was necessary for potassium efflux, and so we're minimizing potassium efflux, so we're minimizing the thing that causes a negative or polarized, so minimizing polarization is a depolarization, so A would work. Glutamate is an excitatory. Excitatory is almost always associated with excitatory. GABA is an inhibitory neurotransmitter that's associated with hyperpolarization. An inhibitor of the sodium-potassium ATPase is also going to minimize the potassium gradient, right, by preventing potassium pumping. Potassium pumping would be reduced, potassium gradient would be reduced, less gradient, less negative resting membrane potential, that would be a depolarization. So if I were to ask which of the following would cause a depolarization, A, B, and D would be the correct answer. Okay, let's take a break. We'll come back to this one. I've got a few more. Think about if you have questions that you want to ask me. Well, you have my undivided attention, and then we'll go on. my pointer. Thank you. Let's go ahead and answer this next one. At the very peak of the action potential, which of the following is or are true?
This one starts to
At the very peak of the action potential. It's a really defined time. The point at the peak. So the short answer to your question is yes, this would be before it declines, because then we wouldn't be at the peak.
Didn't it start declining?
We're at the peak, it hasn't started declining yet. So what's the answer? I think they're all true. They're all true. At the peak, have we reached the equilibrium potential for sodium? Have we gone all the way to plus 66? No. So sodium is flowing in. That's part of why it's depolarized. Because there are voltage-gated sodium channels open. That's how the sodium is getting in, right? This is not in a teaching order, this is in an alphabetical order. Not even that, it's in a random order. A few voltage-gated potassium channels are open. They have started to open, but they are not yet causing the repolarization, because only a few of them are open. If the answer, if this had been many or all, this would have been incorrect, because we're still at a few. The neuron is in the absolute refractory period. At the peak of the action potential, all the voltage-gated sodium channels are open or inactivated. They're one or the other, and another action potential is impossible. The cell is absolutely refractory. So I'm now introducing a concept that you should understand, the mechanism for the absolute and the relative refractory periods.
When you show the potential as like a graph or whatever, when does that actually end? When does the refractory
When
period-
does the absolute refractory period end? It's at the beginning of the after hyperpolarization.
So when it goes below the.
When it goes below the resting membrane potential, now the cell is becoming relatively refractory. And the end of the relative refractory period is when it gets back to the normal resting membrane potential. Now the cell is normal, it's not refractory at all. Okay, is that clear? I could draw a picture. Basically, the simple way to think about it is, during the action potential itself, during the spike, during the depolarization, the cell is absolutely refractory. During the after hyperpolarization, or during the 2 to 3 to 4 milliseconds after the action potential, the cell is relatively refractory. And understanding the mechanisms. During the absolute refractory period, the sodium channels are either fully engaged in the all or none action potential, or they're inactivated. During the relative refractory period, some of the sodium channels have recovered from inactivation, and so we could get another action potential if we hit them hard enough with a large stimulus. Charles?
So the deep is like the dip on like below the resting membrane potential is the relative refractory.
That is associated with the relative refractory period.
Okay. And then once it
Once it
levels-
comes back to the
is
once
the
it comes back to the normal membrane potential, that's the
That's-
end of the relative refractory. Because the sodium channels have fully recovered from inactivation, and the potassium channels have closed, and so everything is back to its normal state. So it's not refractory anymore. Other questions? Okay, we're talking more action potentialology. Which of the following will affect the speed of action potential propagation along an axon from the spike initiation zone, or axon hillock, to the synaptic terminus? Which of these affects the speed of action potential propagation? I'm throwing a curveball in here. temperature speeds everything up. The duration of the relative refractory period does not affect how fast that action potential is moving along the axis. It's following behind the action potential. It's not affecting how fast it's moving forward because it's trailing behind it.
Let me, if you have a thinner axon with signal will propagate faster.
A thinner axon, what do you mean by thinner?
The diameter is smaller.
Nope, a smaller diameter propagates slower. Large diameter propagates fast. Small diameter propagates slow.
Because of the flow of the line.
I could get into the biophysics, but I'd rather not. Okay, which of the following will affect the frequency? How fast action potentials can follow each other. Not how fast they move, but how fast they can follow each other in time. This is how fast they move in space, and this is how fast they follow each other in time. The frequency. This is a challenging one too. I think they're all correct. The greater the duration of the EPSP, the longer the EPSP is, the more likely it is to fire a burst of a train, a series of action potentials. If there's a large IPSP, that's going to decrease so this EPSPs will increase and IPSPs will decrease the frequency. So they both affect the frequency. The longer the relative refractory period, the longer the time must be before the next action potential can come. So that would be a lower frequency. A longer relative refractory period is associated with a lower frequency of action potentials. And a short relative refractory period would be associated with action potentials that can come very quickly after each other, a high frequency. The kinetics of voltage-gated potassium, if voltage-gated potassium channels open and close very slowly, the duration of, voltage-gated potassium channels open very slowly and close very slowly. That means that action potential is going to be very broad because it's going to take a long time to get the potassium channels to cause the repolarization. And then if they close so that lengthening the duration of the action potential would increase the absolute refractory period. And that would change the frequency. Changing how fast they close would change the relative refractory period. And we're back to B. And temperature, like I said, speeds everything up. Okay, let's do a couple more. I don't think there are too many more, so let's just do these. But again, I'm using this to remind you of key concepts. And if you have questions, please, I'm moving through it, but because I'm not seeing hands up, but don't hesitate to raise your hands. And if you want to go back to one of these, if we have time, let's go back. Which of the following are required for neurotransmitter release? So, giving you a hint. Question?
Diffuse release.
Release. We're talking about release. So when I talk about release, I'm not talking about reuptake. I'm talking about release. Exocytosis.
Gonna release just into the synaptic cleft or then the tape of the neurotransmitter from the synaptic?
The release is the release of the neurotransmitter from the presynaptic terminal. I'm not talking about the response, I'm talking about the release. I'm not talking about reuptake, I'm talking about release. That's A and B are the best answers for just talking about release. So this is an important thing, to get the language, we have to speak the same language if you're going to understand what I'm asking on the exam And again, I will be available during the exam, so if you have a question, by release, do you mean the response or do you mean the release? No, I'm using the word release, right? I'm talking about the release. I'm not talking about reuptake, I'm not talking about response, I'm talking about release, that sort of thing. But don't hesitate, if there's a word that you think you might be going astray on, don't hesitate to come find me and ask me a question. You should not have to read my mind. You should be able to understand the question and answer the question. At some synapses, GI-coupled receptors are expressed on the presynaptic terminal. On the presynaptic terminal, GI-coupled receptors are expressed on the presynaptic terminal surface membrane. Activation of these receptors will NT is my shorthand for neurotransmitter. I hope you already know that. Let's walk through this one. What do GI-coupled receptors do? They're inhibitory. Many of them are coupled to a potassium channel that causes, so in addition to inhibiting adenylyl cyclase, many of them cause activation of the potassium channel. That's going to cause, this C is partially true, it's going to hyperpolarize the presynaptic terminal, that potassium channel. But this now it gets wrong. Adenylyl cyclase is also sometimes, phosphorylation of proteins is also modulatory of positively modulatory of neurotransmitter release. So inhibition of adenylyl cyclase and inhibition of protein kinase A will also, I didn't teach this, but that will also decrease neurotransmitter release. But the point is, knowing that an inhibitory receptor is expressed on the presynaptic terminal, inhibition is going to decrease the function of that terminal, which is to release neurotransmitter. What we're talking about here is an axo-axonic presynaptic inhibition. So at some synapses, GABA A receptors are, so this GI-coupled inhibitory receptor is GABA B receptor. This is just code for GABA B receptors. GABA A receptors, which you know are chloride channels. What would be the answer if I said at some synapses, GABA A receptors, ionotropic chloride permeable channels, are expressed on the presynaptic terminal? What would the answer then be? The same. Because inhibition, it would cause a hyperpolarization of the presynaptic terminal. Why would a hyperpolar, but so I'm jumping because I know something that maybe you're forgetting. Why would hyperpolarization lead to decrease in neurotransmitter release? What do we need to get neurotransmitter release? Calcium. Where does the calcium come from? Voltage-gated calcium channels. What opens voltage-gated calcium channels? Depolarization. Is hyperpolarization adding to or subtracting from depolarization? It's subtracting from, so that's why we get inhibition. Which of the following is true for dopaminergic systems? Go ahead and reveal. Okay, what's wrong with A? The locus ceruleus is where noradrenergic neuron cell bodies are. It's the substantia nigra and the ventral tegmental area is the area where dopaminergic neuron cell bodies are located, so the name is wrong. Postsynaptic cells express ionotropic and metabotropic dopamine receptors. What's wrong about that? There are no ionotropic dopamine receptors. Why is C correct? Tyrosine hydroxylase. It's asking you to know that tyrosine is a precursor of all the catecholamines. So I'm using the language of catecholamines. Dopamine, norepinephrine, epinephrine. Catecholamines come from tyrosine, and there's an enzyme. I didn't ask you to memorize the name of the enzyme, but you could have figured out that this is likely to be true based on what you do know. Are catecholamines monoamines? Yes. So no, that was the key bit here, knowing that catecholamine, that dopamine is a catecholamine and dopamine is a monoamine. And monoamines are reuptaked into vesicles by a vesicular monoamine transport. I'm not asking you to memorize the name, but the concept is there. Clear on what I want you to know and what I want you to apply on questions like these. That's key to understand how you should memorize and what you should not memorize, because I know that time is limited. Okay, I don't know how many more of these, but there can't be too many more.
How much should we try to memorize the names of the receptors? Or like just the first one?
I wouldn't memorize names of receptors at all. You should know that acetylcholine has ionotropic and metabotropic receptors, but don't worry about nicotinic versus muscarinic. Is that clear? Which of the following neurotransmitters is essential for a functional sympathetic nervous system? So now I'm asking you to remember the concept of the sympathetic nervous system that we taught last week. The sympathetic nervous system. Remember, we have pre-ganglionic and post-ganglionic neurons of the sympathetic nervous system. Which neurotransmitters are important?
Sorry.
Okay.
That wasn't toilet.
Okay, let's reveal. Acetylcholine and norepinephrine. Acetylcholine is the neurotransmitter released from the pre-ganglionic neuron, and norepinephrine is the neurotransmitter released by the post-ganglionic neuron of the sympathetic nervous system. So yeah, they're both important for the sympathetic nervous system. Which of the following receptors, now we're talking about receptors, are important for a functional parasympathetic nervous system? Remember, we got pre-ganglionic and post-ganglionic. Now we're thinking about the receptors in that pathway. Acetylcholine is the neurotransmitter at the so the presynaptic neuron releases acetylcholine and it binds to nicotinic receptors on the post-ganglionic neuron. Acetylcholine is released by the post-ganglionic neuron and it activates muscarinic receptors on the target of the post-ganglionic neuron. The target of the post-ganglionic neuron, the target cell. So reminding you of key concepts of the sympathetic, the autonomic nervous system. That was a key part of a lecture from last week. Right? So we moved all the way from basics all the way to last week. Maybe we're done. Yeah. Questions about how to prepare, what to prepare, you know, a specific thing that really threw you from any of these clickers? So the book has way more information, the anatomy coloring book has way more information, and the textbook has way more information. Yes, that's true. So use the key concepts list to target your learning for what you need to know from the books. The PowerPoints are also, so if I set it in class, and it's on the key concepts list, it's important. No, I mean, that's helpful to, for me, it helps me to remember the anatomy, but I'm not gonna ask you which is long and which is short. Neurotransmitters and receptors, it means location of cell bodies. Where, you know, for example, the preganglionic cell bodies of the sympathetic nervous system are in the spinal cord or in the sympathetic ganglion. The cell bodies of the preganglionic neurons of the parasympathetic are in the brain, right? We have, because the preganglionic are long, right? So knowing, being able to interpret this picture about where the cell bodies are, where the ganglion is, and where what the targets are. Okay, well, remember quiz 2. I mean, I've been focusing on exam 1 because that's coming up next week, but don't forget quiz 2. Due tomorrow, 4:30 p.m.